Good afternoon. This is Dr. Dan Guerra. I come to you from Authentic Biochemistry Studios, as you can see. And today we're going to do some foundational lipid biosynthesis. Today is the 10th day of May, and of course that is in the year 2021. Sorry, we haven't skipped over this year. It's still here. You can take of that whatever you like. So let's do a slideshow, and let's start from the current slide. Okay, so let me move my thing up here. Get started. Now remember we went through beta oxidation about, quite a while ago, about 3 weeks ago. You know that occurs in the mitochondria. You know that coenzyme A is the acyl thioester group carrier, set up with a fatty acid. Let's say it's an even chain fatty acid. We went through the fact whether or not it was saturated or unsaturated or polyunsaturated. I also talked to you about cis-trans double bonds, all of which, as you recall, can be easily beta oxidized. Hope I set that pseudo-argument straight. Anyway, fatty acyl-CoAs are first oxidized with flavin adenine dinucleotide. You make an enoyl-CoA, which is then hydrated. Then you're left over with the 3-hydroxyacyl-CoA, which then once again gets oxidized, this time with nicotinamide adenine dinucleotide. So because those are the oxidizing agents, they get reduced. FAD is reduced to FADH2, NAD to NADHH+. Then you are left with a keto group, which is the most oxidized that you're going to get for the fatty acid. Then you do a coenzyme A transfer to remove those two fully oxidized carbons, and you have an acetyl-CoA, which is the most oxidized form you can get at this level of solution chemistry. And then that acetyl-CoA, of course, can be utilized in various metabolic modes. One is, of course, that it can be run into the TCA cycle, and that TCA cycle can then utilize acetyl-CoA to make more NADH and more FADH2. However, typically what happens is that the acetyl-CoA is used to make ketone bodies, which are acetoacetate and beta-hydroxybutyrate. And in that process, then you have a soluble carbon, which can be translocated anywhere in the body, makes it into the circulation. It can go to any tissue, including the brain. Remember, the brain discriminates against fatty acids unless they're brought in by certain apolipoproteins and only under certain conditions. So if the brain is going to use acyl carbon, it's going to use it almost exclusively as a ketone body. Remember, that's acetoacetate and beta-hydroxybutyrate. So now, though, we're going to talk about biosynthesis, fatty acid biosynthesis, which is the other side of the slide. Now, biosynthesis in animal systems, not in plants where it occurs in the chloroplast, but in animal systems occurs in the cytoplasm. And the fatty acid synthase is a polyprotein. And that polyprotein will then use acyl carrier protein as the thioester group transfer substrate for all the reactions. And you're going to be using NADPH because fatty acid synthesis is that canonical reductive biosynthetic pathway. So the way that this works out is you get a C2 donor, carbon 2 donor, but you're actually using malonyl-CoA, which everyone knows is 3 carbons. Now, malonyl-CoA was synthesized by acetyl-CoA and bicarbonate in the Acetocarboxylase reaction, which we're going to talk about here briefly and soon. But the malonyl-CoA will be added on to the growing fatty acyl chain, and in so doing, carbon dioxide will be released, and the CoA will also be released because now you're on to acyl carrier protein. You start off with either, if you are already in the biosynthetic mode, you're already attached to ACP, but any incoming, such as the malonyl-CoA, obviously is going to be esterified as that thioester. So the malonyl-CoA, as I just told you, is synthesized from acetocarboxylase. And so biosynthesis is reductive. We don't use NADH, we use NADPH for reductive biosynthesis. You then make the hydroxyacyl ACP, and that is actually a D-beta hydroxyacyl moiety. You then dehydrate and make the enoyl, right? Double bond now is there. And then you just close up that double bond by reducing it with more NADPH. So 2 moles of NADPH are necessary, and there's a dehydration. So there is, again, reduction, dehydration, reduction. And then you are poised at CN plus 2. You've added 2 carbon units. Okay. So these are 2 distinct systems. Beta oxidation is in the mitochondria, and it uses CoA thioesters. Biosynthesis in the cytoplasm, and it uses acyl carrier protein bound to the polyprotein FAS, or fatty acid synthase. Right? So there is a huge metabolic zonation here. One's in the mitochondrion for beta oxidation, the other's in the cytoplasm, that's synthesis. But also you have totally different proteins involved in biosynthesis and degradation. In fact, this was the first time in metabolic biochemistry where this was clearly demonstrated. And when it was demonstrated, it was done so by Acyl, who was the first person to show that biosynthesis and so-called degradation, because that's what beta oxidation is carried out by 2 totally distinct pathways. So it's a dramatic change in our understanding of biochemistry. Now, I've mentioned this before many times in lecture. If you've been watching Authentic Biochemistry and Vera Med lectures, I always do emphasize it, though, because fatty acid synthesis, the study of it, and fatty acid oxidation have revealed a number of paradigm shifts in our understanding of biochemistry. This all started back in the 1950s, late 1950s, and actually is still ongoing here in 2021. So fatty acid metabolism, tightly regulated, we know that. We're going to talk about that as well. But even just the plain biochemistry of its metabolic pathways is something that has inspired a lot of biochemists to pursue biochemistry as a career, myself included. But also, it has revealed a lot about the structure-function relationships of metabolic pathways. And so there's a whole history behind that, and I like to emphasize it. So let's continue on here from this side, and let's look at, this is the phosphopentathene prosthetic group of acyl carrier protein. So this is a protein. So this is a serine residue in the ACP, and that serine residue is going to contain, or is going to possess, this phosphopentathene prosthetic group right here. Okay? So you've got this phosphate ester to this carbon atom, and that oxygen actually comes from the original serine hydroxyl group, because remember the R group of the amino acid serine is an OH. So this entire moiety here is a phosphopentathiene. Now notice it is identical to phosphopentathiene of coenzyme A. In fact, that's where it comes from. And you can see it still has the cysteamine at the end here. And this is actually the important part of the molecule relative to making the thioester, because see, this is the free SH group, either on ACP or on coenzyme A. And of course it is, because the prosthetic group itself, which comes from pantothenic acid, which in, of course, mammals is an essential in the diet, so it's a vitamin, pantothenic acid. But of course, in lower organisms, microbes, and also in plants, you can synthesize the pantothenic acid de novo, right? Actually comes from amino acid metabolism, as one might guess, looking at the structure. So the distinction here is that coenzyme A is a low molecular mass, reasonably soluble, at least in terms of critical micellar concentration. We have to also include that in the discussion of the biophysics and biochemistry of coenzyme A thioesters. But it is a low molecular mass molecule that moves around fatty acids as thioesters. And as we've been saying, is important for the beta oxidation, but it's also very important for biosynthesis because acetyl-CoA and malonyl-CoA are going to be basically substrates for fatty acid biosynthesis and degradation or beta oxidation. But when the fatty acid is esterified to acyl carrier protein, it's exclusively a biosynthetic route. Now, there are a couple, when I think about it, there are a couple of slight caveats to that, and have to do with mitochondrial metabolism and the synthesis of short-chain fatty acids, and also some in the cytoplasm, where they're utilized for covalent modification of proteins. Sometimes ACP is the thioester associated with those fatty acids as well. But we're not talking about that today. We're just talking basic fatty acid synthesis. So I wanted to show you that the phosphopentadiene group is associated both with an acyl carrier protein as a prosthetic group for the protein, embedded within the polyprotein FAS, at least in mammals. And it's also the same prosthetic group that's in coenzyme A. Okay. So this slide here shows you the transfer of acetyl-CoA from the mitochondria to the cytosol that occurs for fatty acid biosynthesis. And so what happens here is a lot of different ways of looking at this. And it has to do with the actual, efficient cause, or that is the actual precursor of the carbon that enters fatty acid synthesis. And so there's some unique phraseology of that in terms of biochemical rhetoric, and we can get into that later. But right now, just look at glycolysis generating the carbon. So you've got glycolysis here, and you know that because of glyceraldehyde 3-phosphate dehydrogenase, and also because of the malate dehydrogenase, you're going to be making some NADH, right? And so that NADH from glyceraldehyde 3-phosphate dehydrogenase in the cytoplasm can be reoxidized via the malate dehydrogenase. So let's start there with oxaloacetic acid being converted to malate. Malate then be converted to pyruvate. So that's one source of pyruvic acid. That enzyme is malic enzyme, and it's decarboxylating, of course, right? So when this occurs, also notice that you make NADPH. So this is one of the major sources of that reducing power I told you and just showed you necessary for fatty acid biosynthesis, right? Now you also get NADPH from the oxidative pentose phosphate shunt, which we've talked about a great deal when we discussed carbohydrate metabolism. Remember, that's using glucose 6-phosphate in that pathway that is also cytosolic, that is not glycolysis, that involves the synthesis of basically C5 and C7 carbohydrates that is linked to the final utilization of glucose 6-phosphate after decarboxylation, and of course its oxidation to generate 2 moles of NADPH, right? So these are the glucose 6-phosphate dehydrogenase and 6-phosphonoglucanolactone dehydrogenase. Those two dehydrogenases in the oxidative pentose phosphate pathway also contribute excessively and immediately to the production of NADPH necessary for fatty acid biosynthesis, which is in cytoplasm, where the oxidative pentose phosphate shunt and the malic enzyme are all operating. So anyways, again, looking where OAA comes from, now watch this. If you've got pyruvate being synthesized from glycolysis, which you know is a major end product, pyruvate can enter the mitochondrial matrix and go through the pyruvate carboxylase reaction or the pyruvate dehydrogenase reaction. We've covered this extensively just a little while ago in lecture. Now, notice that when you condense these two precursors to citric acid, OAA and acetyl-CoA, with citrate synthase, you make citrate. This is all happening in the mitochondria. Now, rather than citrate continue on the tricarboxylic acid cycle, if there is adequate amounts of NADH, the dehydrogenases in the TCA cycle will be filled up plenum with NADH, right? And so that will block the citrate, the isocitrate dehydrogenase, which is the first oxidizing enzyme in the TCA cycle, and after citrate is converted to cysticonatate and then isocitrate. And so if you have adequate amounts of NADH, you won't run the isocitrate dehydrogenase in the mitochondria, which means the citrate can then be bulk transported out of the mitochondria, as shown here. And as it moves out, because it's a carboxylic acid transporter, right? Pyruvate moves in. Pyruvate moves in as citrate leaves. Now watch what happens when citrate leaves. It comes up to an enzyme, cytosolic, called ATP citrate lyase. ATP citrate lyase utilizes ATP to carry out that carbon-carbon bond cleavage, and it resynthesizes now in the cytoplasm acetyl-CoA and OAA. We just saw the fate of OAA, right, in this canonical sort of descriptive cartoon. OAA can be converted to malate via the malate dehydrogenase, right? And in so doing, utilizing the NADH to synthesize from glycolysis in the glycerol-3-phosphatidehydrogenase pathway, resynthesizing NAD so the glycolysis can continue, right? At the same time, making malate, which can be converted to pyruvate. So the pyruvate moves back in. So that's one fate of OAA, okay? Notice, though, the other fate of carbon from citrate is acetyl-CoA. The acetyl-CoA, of course, will be the precursor for the acetocarboxylase, which will make malonyl-CoA, and then the malonyl group will be transferred to the fatty acid synthase, and then you'll be able to now couple this mitochondrial metabolism with cytoplasmic metabolism at the same time running glycolysis to be lipogenic, meaning, of course, you synthesize fatty acids. This is where you get the NADPH from, some of it anyway. I already mentioned the oxidate pentose shows a very, very significant contributor. But as malic enzyme, I'm just showing you how the pathway can be, the circuit can be completed, right? But also at the same time, you're synthesizing fatty acids and you've got the NADPH to do it, right? Okay. So let's continue on. So here is the carboxylase enzyme. It takes acetyl-CoA and bicarbonate, and it utilizes the hydrolysis of ATP, and it makes this malonyl-CoA right here. Right? That's malonyl-CoA. Now that carboxylic acid is going to be lost, right, as you saw, almost immediately during the fatty acid biosynthetic pathway. But you synthesize this malonyl-CoA, and that's going to be one of the initial precursors to triggering the initial sets of reactions for fatty acid synthesis, right? So this enzyme has been well studied, also by the same great lipid grandfather, Salih Wakil. He studied the acetocarboxylase back in the late 1950s in some really elegant, excellent papers that were published that long ago. First time that was shown what biotin was necessary for in the cell, because it's a biotin-containing enzyme. You make a carboxybiotin as an intermediate in the carboxylase. The carboxylase is well regulated. There's actually two forms of ACCase. One of them is associated with the mitochondrial membrane, and it becomes inhibited by mal-CoA, which is the product. And in so doing, right, what that does when you block that pathway, you also block because you're in the mode of making fatty acids. You are blocking the beta oxidation pathway because that carnitine palmitoyl transferase is inhibited because of that interaction with the acetyl-CoA carboxylase. It's mitochondrially membrane bound and associated, right? So the carboxylase itself, though, is also turned up, that is allosterically regulated, and its rate constant changes because of the cytosolic production of citrate. So citrate will actually cause the acetocarboxylase to be more active because it causes the acetocarboxylase actually to polymerize. So rather than having a monomer of the enzyme, which will still catalyze the reaction but at a much lower rate, it polymerizes into rods in the cytoplasm, and it's a much more active enzyme. It becomes basically a molecular biosynthetic motor to make a lot of malonyl-CoA for fatty acid synthesis. Remember, fatty acid synthesis is absolutely integral, vital, essential to every cell. because you can't do anything unless you make fatty acids. Now, why is that? Because you have to make membrane. So as DNA synthesis is going on, the S phase, for example, right during cell cycle, right, you're also mobilizing this entire pathway and synthesizing fatty acids. That's why lipid metabolism is so closely linked to cell cycle. Cholesterol as well, because the acetyl-CoA you make in the cytoplasm for fatty acid synthesis is also equally used for prenolipid biosynthesis via the isopentyl pyrophosphate pathway, or also known as the mevalonic acid pathway. And that will be for the synthesis of sterile lipids. Remember, you have two different major classes of lipids. You have acyl lipids, fatty acids, and you have the prenolipids, major component of the membrane and also precursor to sex hormones and mineralocorticoids and corticosteroids is, of course, cholesterol. And also that totally impacts membrane fluidity changes and water movement through the membrane. So cholesterol is really an absolutely essential membrane prenal lipid, right? As well as what we'll find out later, the glycerolipids and the sphingolipids are. That's what makes up the membrane. Okay. So went through this very briefly, but this is the carboxylase, very important. It's basically the rate limiting step for fatty acid synthesis, although it's not part of the FAS complex because it synthesizes mal-CoA, the precursor for the FAS. Let's continue. Now here's the enzyme itself, just giving you a little bit more detail from these slides from textbook. Right? So you've got this biotinylated enzyme, so it has a biotin moiety covalently bound to it. It picks up water-solubilized CO2 or bicarbonate, and again, with the hydrolysis of ATP, you make that carbon-carbon bond. The CO2 now is acting as a carbanion. So you have a carboxy biotinylated enzyme as an intermediate, reacts with acetyl-CoA, and there's malonyl-CoA, and then you release free enzyme with its prosthetic group biotin, right? All right. That's just another, so I spoke this, and there you see it in a little bit more detail. I'm not showing you the structures there. We could do that later. Here's some structures for you, though. Let's go through fatty acid synthesis. So here's this acetyl-CoA, and we know where that comes from in the cytoplasm, which I showed you, HP citrate lyase, right? And it's going to react with the reduced acyl carrier protein embedded in the fatty acid synthase polyprotein in the cytoplasm, right? So this reaction here, acetyl-CoA and ACP, right? It's going to be the acetyl-CoA ACP transacylase. It's also known as the malonyl-CoA ACP transacylase because the same reaction you see here will take a malonyl group, right? So it could take a malonyl group or it could take an acetyl group. When you first start out, though, with acetate, even before you make malonyl-CoA, you can actually start the reaction, the fatty acid reaction, by making the acetyl ACP, right? Now, at the same time, the malonyl-CoA, which was synthesized from the acetyl-CoA, will likewise transfer out its coenzyme A thioester for ACP. So now you have acetyl ACP and you have malonyl ACP, right? Those are the structures right there. Remember thioesters. And so this condensing enzyme, it's known as the beta-ketoacyl ACP synthase, will now take acetyl and malonyl ACP and make acetoacetyl ACP, notice, but it'll also release the CO2 that was originally trapped by the carboxy biotinylated acetyl-CoA carboxylase, right? So this actually drives the reaction, this CO2 release, right? So this is basically an oxidation, right? That's what's going on. And so this, the acetyl ACP, though, is actually, the acetate is actually going to be transferred to a cysteine residue on the fatty acid synthase complex, right? So the enzyme itself, the beta-ketoacyl ACP synthase, is now carrying that acetate. So it started off as a part of the ACP moiety, but it's swapped off of the ACP, all in the backbone of the fatty acid synthase, and transferred to a cysteine residue now, a cysteine residue now, on this enzyme, the beta-ketoase ACP synthase. That's what that sulfur is there. That's actually from a cysteine residue on the enzyme. Still thioester chemistry all the way. So that's how you don't have two competing ACPs. That's why you don't see ACP as a product here, right? Because you've already cycled that ACP back, right? So it can pick up a new acetate. Understand the real beauty of fatty acid synthesis. It's just an absolutely glorious biosynthetic pathway, all happening again as a polyprotein. So now you've got acetoacetyl ACP, right? That's exactly what it looks like right there. It's thioester of ACP. You start reducing, right? Because you got this keto function. So the first reaction, again, using NADPH, either from the oxidative pentose phosphate shunt or from that malic enzyme, all cytosolic, right? That NADPH now is going to be used for this reductive biosynthesis. And there you have now this D-beta hydroxybutyryl, which it's only the 4 carbon, because I'm showing you from the beginning, acyl ACP. So this enzyme here is beta-ketoacyl ACP reductase, KR for short, right? And you're going to then oxidize the NADPH. And so that's going to have to be resynthesized. There's the NADPH going to be resynthesized. And it's easy to do because you're running that whole OAA, right? From the release of OAA and acetate from the citrate that's entering the cytoplasm. Because all that carbon that was in the citrate, for example, from glucose, you see how glucose is converted to fatty acid. That's what's going on here, right? It's exactly what's going on here. Right? Sure it is. All right, so what happens now is you've got this D-beta hydroxybutyryl ACP. That's the product of the reaction. Now you're going to dehydrate. You're going to get rid of water. So there's one OH. You're going to take a hydrogen off that carbon there. See? And now you're going to be left with, alpha-beta transbutenoyl, because you got a double bond there, right, between this carbon and this carbon, right? So there's a carbonyl group. And then this is the alpha-beta structure here we always talk about, okay? And now notice it's a trans double bond. It's a transient trans double bond because it's going to get immediately reduced because it's on the backbone of the fatty acid synthase by now this enoyl ACP reductase. It's a part of the polyprotein. It's another enzyme that's embedded in this polyprotein. It's fusion of all these enzymatic reactions, right? Okay. And we'll talk more about the genetics of the FAS later. Right now, I'm just doing straightforward biochemical pathway. So notice now this enzyme will also utilize NADPH. Again, you come from the oxidative pentose phosphate shunt from the malic enzyme. And now you've got this fully reduced butyryl ACP. 4 carbons, completely reduced. Remember, fatty acids started off as completely reduced, saturated fatty acids. And the end product, the ultimate end product, is actually going to be palmitic acid, C16:O. 16 carbons, no double bonds, right? That's got to be the end of it, shown down here. Okay, so now the same thing that happened with these all series of reactions here that are going to continue. And 7 reactions later, because you have to build up the C16, right? You're adding 2 at a time. You already got this 4 moiety here. You're going to end up with palmitoyl ACP. And then part again of that fatty acid synthase, part of that whole polyprotein complex is this enzymatic reaction, palmitoyl thioesterase, or just the thioesterase. Passes water over that thioester bond, and you're left with palmitate, and you're left with free ACP. And you go all the way back up here, and you can see the reaction starts over, right? So there's a competition as the fatty acid chain is growing, moving that carbon around from that cysteine residue on that initial condensing enzyme back and forth from the ACP, right? So you can get some slowing down of fatty acid synthesis if indeed your thioesterase becomes rate limiting, because you have to release the end product. Now you can also release shorter chain saturated fatty acids. For example, myristic acid, C14, right? You can even release octanoic acid, which is a C8 fatty acid. And that's really important because you have octanylation of proteins. One that comes in mind is ghrelin, the adipokine ghrelin, right? Which is actually a feeding stimulant, actually works with growth hormone. When you octanylate ghrelin that goes to the brain in the, remember, the arcuate nucleus of the hypothalamus, and that then triggers the feeding response, right? Because you've utilized that 8-carbon fatty acid. So you don't always make palmitate, but almost always. And there are unique thioesterases which will give you shorter chain fatty acids, such as myristate, such as dodecanoate, and the octanoate. And those are utilized for totally different purposes in the cell. So it's not always just net biosynthesis of fatty acids. So understand all the pleiotropy involved here. Okay, let's continue on here. So again, this is showing you the same reaction mechanism. I'm just showing you a little bit differently so you get the idea of the individual reactions. There's acetyl-CoA, there's ACP, there's acetyl-ACP being synthesized. Here is the malonyl transferase reaction. Here's malonyl-CoA and ACP. So now you've made malonyl ACP, right? Acetyl-ACP, malonyl ACP. Here's your condensing enzyme, right? Here is the acetyl-ACP and the enzyme itself with that cysteine residue. And here you've made now the acetylated enzyme and free ACP, so it can go back in and pick up more acetate or more malonate. And then the condensing enzyme itself then will get involved in the reaction, taking the malonyl ACP and its acetylated form, and the enzyme reaction will be catalyzed. And now again, you're left with this C4 intermediate. You've lost the carbon dioxide from that malonyl group, and you've got the freely released condensing enzyme to do another round, right? And then you can just see the same reactions we just talked about. There's the beta-keto ACP reductase, right? There's the hydroxyl group. You got to dehydrate that. You got to get rid of that water. You do that, you're left with this trans double bond. It's actually trans, doesn't show it there, but it is indeed. And you do that second reduction, and then you keep on building that way on the ACP as the thioester backbone. Then you just move that over now, right? You'll move over that growing acyl chain to that condensing enzyme each time. So you're always staying in thioester chemistry. Okay. Amazing, man. So again, it's just showing you the end product here after 7 reaction cycles. C4, you get to palmitate, you get thioesterase. Now, there's palmitic acid. You never have free fatty acids in the cell. So you're going to use that palmitate immediately to make palmitoyl-CoA. And once you make palmitoyl-CoA, what you're going to do is make glycerolipids or sphingolipids with it, right? Or you can also elongate the palmitic acid to stearic acid, that's C18:O, and then you can make an initial desaturation, making C18:1 delta 9, which is also known as oleic acid, the most common monounsaturated fatty acid in the de novo fatty acid synthesis pathway, right? We haven't gotten there yet, but that's basically what can happen with beta palmitate then, transferred into glycerolipid biosynthesis or sphingolipid biosynthesis. So part of triglycerol, part of glycerolipid biosynthesis is phospholipids like phosphatidylcholine, phosphatidyletherine, phosphatidylmethylamine, phosphatidylenoacetol, phosphatidyleglycerol, and of course cardiolipin, which is diphosphatidylglycerol. But you also then can utilize those fatty acids to make triacylglycerol, and you can convert that to diacylglycerol, which can turn on protein kinases C, right? So there's a lot of metabolism we've talked about before, signaling metabolism linked to these de novo pathways. But ultimately, that palmitate can be converted just to plain old triacylglycerol, loaded onto a lipoprotein. This is happening in the liver, of course, and then that trafficked to the adipose, and then laid in as visceral fat, right? You see how this is all linked together. Now, all the hormonal regulation, all the enzymatic regulation, and all the heavy lifting that's carried on by allosteric modification of the enzymes involved in these biosynthetic pathways are all working in concert to make fatty acid, to make membrane, and to make carbon source to derive ATP because of the beta oxidation pathway we talked about last time on the video, right? So you see how this all comes together, right? This is the biosynthetic pathway. And it's really important that you understand it's unique and distinct from the beta oxidation pathway. All this is happening in the cytoplasm. So you have no problem taking that palmitate then and running it, burning it right back through beta oxidation, because you blocked off that carnitine palmitoyl transferase system because of the buildup of malonyl-CoA. Right? Malonyl-CoA is building up because you're making it from the carboxylase, because you're running the acetate out of the mitochondrion via that citrate lyase reaction, running citrate and then making the OAA and, of course, the acetate in the cytoplasm. There's the thioesterase, as we showed you before. So here's basically the enzyme itself. I told you it's a polyprotein. They're all the individual enzymatic machinery. There's the acyl transferase. There's the condensing enzyme, right? The ketosynthase. There's that cysteine residue I told you about, right? Here's the enol reductase, the ketoreductases. There's the thioesterase, right? So you have an alpha chain and you have a beta chain, okay? So it's a polyprotein. So it's actually a dimer of that polyprotein, all in the cytoplasm, right? Now you can make fatty acids another way, and this is now a ketide pathway. And I'm going to stop here because it's a nice transition, because once we get to the end of fatty acid biosynthesis, just making palmitate, as I said, the other key feature is then going to be actual frank lipid biosynthesis, which is going to involve the glycerol lipids and all the other components I just have been describing to you verbally. And I want to be able to get into that. And I also want to show you that ketide synthetic pathway, because it's an interesting way of synthesizing fatty acids. A totally different enzymatic complex. And your professor here has studied both those ketide synthase pathways and the fatty acid synthase pathways in the laboratory, in my laboratory. All right, so we're going to stop there, and we're going to continue on with this. It's a little bit shorter than usual because I really want you to get the full breadth of this. This is basic biochemistry, right? This is pathway biochemistry. You need to understand this before we move to the next level, which is going to be from fatty acids. We're going to start building lipids, and those lipids are going to start then coalescing into membranes, which is another entire fascinating field of fatty acid metabolism. These fatty acids, once they're esterified, usually as oxygen esters in the various lipid moieties, then can spontaneously form monolayers and bilayers, and also hex phase 2 layers, and sometimes inverted layers of membranes, which will then allow for what? The closing of the cell, and then the ability for the cell to be autonomous and to regulate flow, what is external and what is internal within you without you sort of metabolism. So all of this is linked, of course, to cell cycle, because as I mentioned to you, you have to make new membrane every time you get a cell division. So you need cholesterol biosynthesis, you need fatty acid biosynthesis, as well as DNA replication. So these are linked together. Many of the enzymes I just told you about, and much of that machinery and the allosteric regulation are co-regulated with nucleotide metabolism as well. So I'm going to leave you with that. And I'll just tell you that, again, this is Dr. Dan Guerra from Authentic Biochemistry Studios in the beautiful Inland Pacific Northwest. I'm looking outside, and I'm not going to turn the camera around, but the skies are blue. The clouds are that artist sky, like cumulus, with a little bit of cumulus nimbus in the background, and a green leafy maple tree and a willow and an orchard in front of me. So just absolutely beautiful. In fact, it's so nice I'm going to go outside and take a walk. So take care, and I will see you soon. And of course, I have to say bye for now.